Thank you, Gordon, and also thank you to the organizers for inviting me to this prestigious precision medicine meeting. And I'm conscious that I stand between Gordon and his beer. And so I will start simply that from a distance, all snowflakes are said to be snow. And from a distance, all patients are said to be cancer. But I'm sure you'd agree with me that within snow, there are trillions and trillions of different snowflakes. And so it is with just one case that was published with metastatic breast cancer, a single semantic terminology, 57 consecutive patients, and no two patients had the same mutation profile. And clearly with now 10 to 20 million variants that have been identified, it's probably safe to say that no two patients with cancer have exactly the same profile. And so quite clearly, as is the theme for this conference, we're moving into an era where we not only stratify to try and pull out populations, but also to personalize where we try to pull out the individual. We've gone from giving drugs to all of the grey people in its trial by therapy to where we can put the molecular microscope against people and inform the type of treatment that they are to receive. Now that all sounds very easy to say, and the purpose of this talk is to tell you about the UK experience, which you may think is straightforward, but there are 6 challenges that I'd like to describe from a trial that we've set up in the UK because it's a trial. It's not commissioned in the UK. It's not available as a test on the National Health Service. And so the trial that I'm about to describe and the challenges embedded within it take the patients and we profile them against both the tumor tissue and also against their tumor material circulating in the blood. We sequence those, we bring them together through a molecular tumor board, we then decide on treatment. As I say, it's easy to say, and I'll walk you now through 6 challenges that we've faced over the last 3 years in proving this into a system which is now relatively straightforward for patients to access. So the first, and I've not heard this spoke about so far today, is to consider what ethical position you want to take before starting this endeavor. And in this regard, we found the University of Michigan's publication really very informative to try and unravel what clearly is a highly complicated area of genetic testing into something which is as straightforward as 2 sheets of paper on an informed consent. And to start off with the first bucket, the flexible default model says, look at your cancer of interest, and if on the first row there's a treatment available, then that's a non-flexible choice and you disclose that to a patient. If, however, you find a result on the second row that is at increased risk of cancer for the patient, then that's flexible in the consent in terms of whether patients do or don't want to know that information. And if the mutation is unknown, of unknown consequence, again, that's not flexible and we don't disclose it. But what if you pick up conditions in your genetic profiling that are not relevant to the cancer that they're being profiled for? And again, there are a couple of options. If you have clinically relevant disease, a mutation where we know that there's relevance to the disease beyond cancer, then the patient has a choice flexibly for that to be disclosed. And again, they have the flexible choice if you find something of biological relevance for the family, again, for that to be disclosed. But similar to the cancer, if there's no known understanding of that mutation, then it's not flexible and it's not disclosed. As I say, we took the consent to Independent Cancer Patients' Voice, which is one of the patient groups in the UK, reduced it to a 2-page consent form that is now published, and if you guys, in setting up these things, want to have access to that consent and a 10-page patient information leaflet that accompanies it, then very happy to share that with you. It condenses about a year's work going through ethicists into a very straightforward publication. But once you've sorted for the ethics, there's a slight administration problem, as Giuseppe referred to, which is you need to get some very disparate and very busy individuals, medical oncologists, clinical geneticists, bioethicists, all in the same room, as we do every month for an hour and a half, to look after about 15 to 20 patients worth of data and to make one of 3 recommendations: to either advance the patient to a clinical trial, to actually nothing available, or to conduct further translational research, particularly PDX, patient-derived xenograft models, or maybe CDX, clinical trial, circulating tumor cell-derived xenograft models. And as I say, that all sounds very straightforward, and it was for a while when we maybe only had 1 or 2 patients each month, and we looked at their paper copies of their tumor DNA and their paper copies of their circulating DNA and their paper copies of the clinical data. As that molecular tumor board started to disperse itself across Manchester and the globe and couldn't attend, and the volume of data got greater, we started to put it onto a web-based database platform where each month we can look at these wherever we are in the globe now and bring together that disparate expertise for an hour and a half each month. And it's likely that we're going to have to move to an hour and a half each fortnight given the volume that we're now moving into. Once you've solved for the ethical and logistical challenges of how you get these data reviewed, there's the technical challenges which often populate precision medicine talks, so I won't dwell much on this, save to say that we use a homegrown tumor panel, tissue panel in Manchester using the Illumina MiSeq platform. We look at only 24 genes with this platform that requires a 20 nanogram DNA input, and the genes are there on the right-hand panel. We also have the luxury within Manchester of having access to foundation medicine tests through the collaboration that we have with Roche, and we've now profiled over 200 patients through the 315 gene panel based upon tissue, and are due to publish our concordance figures later this year between the foundation test and the home brew test. The circulating free DNA panel, we look at 653 genes now through the Illumina NextSeq 500 platform that requires a 5 nanogram DNA input and has a limit of detection of 2.5%. Now, I don't intend you to read this slide for one moment because it's the data for the first 100 patients that we profiled in 2015 and 16, but let me color code it for you. In deep blue, we have an exact match between the tumor and the circulating tumor DNA. And in the light blue, we have a match, but with some additional material that either tumor or blood has thrown up. And you can see on the right-hand sort of heat map that the vast majority of samples, there is good concordance between circulating tumor DNA and circulating blood, resonating with the previous speaker. I was privileged when part of AstraZeneca to work on the T790 test, which was the first test, because of that good concordance. There are some discordant samples in purple, but they're quite rare. And so overall, the concordance rate is 80% for the actionable mutations, the 13 that we call actionable at least, based upon the platform that we're using. And those are the ones that we saw in green. Having set up your molecular tumor board and got your way through ethics committees and done the sequencing, the wicked problem that we're now facing is the unknown unknowns, and even the known unknowns, where the science hasn't yet kept pace with the variant calling. And again, this was referred to in brief by Giuseppe. There are numerous databases now where we can look at something that we believe is signal rather than noise. But then the question is, how do you translate that signal into a clinical trial that's available? And from about a dozen or so platforms, we've settled on 2, one by Fabric Genomics and the other by QIAGEN QCI. And we're trialing those 2 pieces of software, also alongside the IBM Watson software, to get at the fact of no one can hold in their mind, not even Jeff, bless him, the 10 million variants that these things throw up, and whether they're linked to a clinical trial or not. And so again, we're using bioinformatics and software engineers to try and do that matching to clinical trials. Fifthly, there are organizational challenges, and again, our previous speaker referred to this in his talk, that it's one thing to recommend a trial, it's another thing to actually get them onto the trial. So these in the left-hand panel are our first 100 patients tested. We found 41 that we had an actionable mutation, one of those 13 mutations that I showed you on the previous slide. But of those 40, only 1 in 4 got onto a clinical trial. And that's because even at the Christie, with 50 open trials right now in phase 1, we don't have a sufficient breadth of clinical trials to match these patients onto. And so we've gathered together with 5 other centres in the north of the UK, Newcastle, Belfast, Leeds, Sheffield and Liverpool, to call ourselves the Experimental Cancer Medicine Centre North Network, simply to double the numbers of clinical trials that are available to our patients. Later this year, I'm excited because for the first time in the UK, Cancer Research UK will release a web-based tool that will allow us to see any centre, all 18 phase 1 centres across the UK, and the open trials that they have. So we'll probably go from having 100 clinical trials at our disposal to about 300, 350 clinical trials to potentially match. So maybe, just maybe, those 41 patients that had the actionable mutation will get maybe a half or 3/4 of them onto a clinical trial because we'll have better visibility of that. However, of those patients that we did match onto a clinical trial, our response rates improved from a lousy 0% in 2012 to running actually just north of 20% when we last audited it last year in 2017. And then there's a financial implication, because as you heard me say before, unlike the US, where they get these tests essentially on insurance, we don't at the moment commission this as a test in the UK, and so we have to find research funding to do the trial that I've just walked you through. I'm very grateful to Ian Walker of Roche for this slide that he showed at a recent board in December, where he collected together the meta-analyses essentially from phase 1 studies published in JAMA, phase 2 studies in the Journal of Clinical Oncology, the phase 3 studies in the JNCI, and also clinical practice studies in Lancet. And irrespective of which one you look at, the phase 1, for example, were looking at whether you match or not, has increased the historic response rate of 5%, up to our own figures now close to 30%, or progression-free survival doubled, or phase 2 studies where the response rate has gone from 10 to 30%, and again you've had a doubling in progression-free and even overall survival, or phase 3 settings where the response rates have gone up from 23 to 50%, and again you've had a roughly 50% increase in progression-free and overall survival. There's a consistent message here. And we'll be taking our data along with data like this to the NHS commissioners this year to see if we can move tests that I've just described to you onto a platform where we no longer have to fund this through charity funding or research funding, but can actually fund it as part of a patient's NHS piece of work. And so at this stage, I just want to show you, I can't take you all to the Christie, but I can bring the Christie through video to you, what that really means in terms of patients' experience on the target study at the Christie. If we can show the video, please.
I'm Dr. Matthew Krebs. I'm a consultant here at the Christie Hospital in the Experimental Cancer Medicine team and a senior lecturer at the University of Manchester. In the world of drug development, a lot of the new medicines that are coming through are no longer chemotherapy, which often are quite toxic and attacks all cells in the body and get lots of side effects. It's much more about targeted treatments. So it's about understanding something about the biology of the cancer, what's causing an individual's cancer, and in particular looking at faulty genes. And a lot of the new medicines are targeted against very specific faulty genes, for example. So really the target trial was born out of that idea. So what we need to be able to do is screen for a whole range of faulty genes all in one go, using as little tissue as possible, so we can screen for a wide range of things and then look at which studies might be most suitable for our patients depending on what we find. And actually, if we can do this from a blood sample, we'll be one of the only sites in the UK and certainly in the world that is capable of doing that, and will be a fantastic opportunity for patients.
Well, I'm Lynette and I'm here today to sign up for the target trial at Christie's. The trial was really interesting to me because I've just gone through chemo for 3 months and it hasn't worked for me, and this sort of trial will give the information that might make it a lot more targeted in future. So I may not have had to have gone through 3 months of chemo, I may not have lost my hair. So I'm really interested because of the database that it's going to create really, and really inform future decisions on targeted treatments.
So Caroline Dive, I'm the scientific lead for Manchester's Experimental Cancer Medicine Centre. I also run a large biomarker science team at Cancer Research UK Manchester Institute, and the laboratory works really closely with the clinicians at the Christie Hospital. One of the things that has become increasingly exciting in the field of cancer research is the ability to measure circulating tumour DNA in about 10 to 20 mils of a patient's blood sample. And circulating tumour DNA is released from cancer cells when they die. So as tumours are growing in patients, some of the tumour cells will die and release fragmented bits of DNA into the bloodstream. And this is particularly interesting when a patient may have multiple tumours. Getting a biopsy from the tumour can be difficult depending on where that tumour is sited in the patient's body, and if it's very inaccessible, obviously it's more challenging. And if the patient has more than one tumour, which one do you biopsy?
The cancer returned on one of the scans, and they suggested, have you ever thought about the clinical trials? So go away and think about it and come back. So we decided to go ahead and do it. And then we got the phone call after they got one of my samples from the local hospital, and it was showing this gene P10, and that's where it came from.
Prognosis originally, Carol was given an average of 12 months survival. 22 months down the road, she's looking well. What treatment she's had is it's obviously working, and this is the next step.
Sometimes we might find faulty genes where we've got a very clear clinical trial available for patients. There might be other circumstances where we find a faulty gene, but we may not necessarily have a clinical trial available for that patient, or a matched clinical trial for that particular fault. So what we'll do in that circumstance is have a look, first of all, across the whole of the northwest region, so ECMC North, to see if there may be any clinical trials available within the region.
Manchester is one of only 2 major centres called out by Cancer Research UK, and it is known for its precision medicine. And over the last few minutes you've seen the way that we can now understand the molecular basis of an individual patient's tumour and match them to a clinical trial. And that precision medicine, as we call it, is pioneering work that we're doing here in Manchester, and is becoming world leading. And hopefully later this year, together with our vanguard partners, the Royal Marsden and University College London, we're hoping to convince the UK government to, as I say, move these away from research tests to tests which are commissioned in the NHS. But just in closing, hopefully I've convinced you, if nothing else, that to embed this requires team science. No one has all of these expertise, and that came home in spades to us last year, in fact 10 months ago, when we had the fire at the institute that Caroline works at, CRUK Manchester Institute, where our circulating tumour DNA was being analysed. And immediately that had a huge impact in April on our recruitment rates to target. But again, the team rallied around, moved the laboratories down the road to St Mary's Hospital that does a lot of the Downs genetic testing for the UK, and re-establish recruitment rates. And it's to that team that really I just need to credit this exercise. So thank you.